So the second thing I wanted to do today is to give a general overview or a review of the life cycle of a virus. All right. So we'll most likely go back and talk about these events in details. But since it's been a while since you did virology back in biology, I just want to remind you about of a few things with that have to do with viruses. All right. First of all, is the idea that a virus is not it's not really considered a living thing. All right. So this right here is an illustration of the coronavirus. And one thing that about viruses is that they are, by comparison to human cells, they are extremely simple. All right. And one reason that they're very, very simple is that they don't have to they don't have to contain all the machinery that's necessary for the replication. All right. For example, they don't need to essentially be fed anything. They don't have any energy requirements all by themselves. All right. So if you had a virus and it's in an environment that's good for the virus to survive in, technically it would never really need food or anything to sustain its life. And the reason for that is that when it does, so the reason that our cells need food is that we need energy in order to conduct all the functions involved in replication of our cells. All right. So why doesn't a virus need that? Well, it's going to, when it infects a cell, it uses the cell's machinery, all right, to do all those functions for it. So it doesn't need to have any of those functions itself. All right. So, first of all, different viruses. There's some things about viruses that are going to be different, but there's some things that are the same. So some differences between viruses. First of all, the outside appearance. All right, we're talking about the coronavirus here, but there's some viruses that have completely different appearances. All right, all viruses are going to have some form of genetic material in their center. All right, that's generically just referred to the viral genome. All right, for some viruses that's going to be RNA. For some viruses it's going to be double-stranded DNA. And just for for the coronavirus, it's single-stranded RNA, and that's going to have an effect on what it does once it's inside the cell. All right. So the first thing that any virus needs to do is it needs to attach to a host cell. All right. So, for example, this would be your virus right here, and this would be a host cell. All right. A lot of times we think of these as being some sort of animal cell, but viruses can also infect plant cells. So it can be a lot of different things. But the first thing that has to happen is you need to bind to that host cell, and you need to find some way to get your genetic material from that virus into the cell. All right. So that can either happen by just binding and inserting the genome into the cell, but sometimes the virus kind of essentially becomes incorporated into the cell. The details of that aren't quite so important, because the end result is that this genetic material, the viral genetic material, ends up getting into the host cell. So now we have the viral genome inside the cell. All right, and in this case, that's going to be considered what's called the positive strand of the RNA. All right, the positive strand is the strand of RNA that is inside the virus. All right, it's essentially that positive strand is going to serve two functions. First of all, it's going to code for all of the proteins and necessary components necessary for it to make a copy of itself. But secondly, it's going to serve as a template, to make other copies of itself. So, it's number one, it serves as something that, it needs to make another version of itself, but it also needs to make the machinery that's necessary to make that copy of itself. All right, and there's some other things that are going to be involved. So after that that strand gets into the cell, it's going to use one of the ribosomes from the cell. All right, so this ribosome right here, it's not something, you'll notice that there's no ribosome in the virus. All right, it has no need for one because it just uses the ribosome of the cell that it infects to do that function. All right, so one of the first thing that happens is the ribosome, and ribosomes, if you recall, are the parts of the cell that make proteins, or they read the genetic material and they make proteins from that code. All right, the first thing that it makes is this virus polymerase. All right, and so the purpose of the virus polymerase. So the first purpose of this virus polymerase is to read that single strand, that positive strand of RNA, and make a negative strand from that. All right. So it's not making a copy of itself. It's essentially making the mirror image. All right. And if you recall from biology, there's when you have the double-stranded DNA, the opposite strand contains a complementary nucleic acid. For example, adenine was always directly across from a thymine. All right, cytosine was always across from a guanine. All right, so essentially that's similar to what's going on here, except for when this negative strand is made, it doesn't make a double helix with the original positive strand. All right, it just makes a single-stranded negative version of the original positive strand. All right, so that negative strand is going to do a couple of different things. One of the things it's going to do is parts of it are going to be read by the virus polymerase in order to make some proteins in the cell that are necessary for the virus's replication. All right, so for the virus to go on and make a copy of itself, it's got to start making the components that it needs for the future viruses within the cell that it's infecting. It can't do this out here outside of the cell. So it's going to takes that negative strand, and it uses sections of that negative strand to code for some of the proteins that it's going to need. So first of all, a little correction of something that I just said. Right, I mentioned that one of the things that the negative strand did was it would start coding for proteins and that were needed for replication. Actually, what it does first is it makes sections of it get read, which get turned into sections of the positive strand. All right, so first we get an entire negative strand that gets made, and then sections of that negative strand get code for what essentially would be small parts of this original positive strand. All right, so instead of making it a full positive strand from this full negative strand, once that negative strand is made, the viral polymerase will essentially make small sections of the positive strand. All right, so essentially this small section of positive strand would just be like a section of this. All right, those sections of the positive strand of the RNA are what are called some subgenomic particles. All right, and those get read by ribosomes to make proteins that are necessary and that are going to be needed in the new virus. All right, one other thing that happens with the negative strand is it'll make a full positive strand from it, and the purpose of that is that it's going to become the full positive strand that's going to be in the middle of the newly formed virus particle. All right, so these white little dots right here, those represent some of the proteins that were made when the subgenomic section of the positive strand makes some of the proteins. All right, and those are gonna combine with the newly synthesized positive strand, which got made from the negative strand over here, and those are going to form what's considered a nucleocapsid, and that's going to essentially bond. This right here is what's called the endoplasmic reticulum of the host cell, and it's what's going to happen. So that endoplasmic reticulum has its own phospholipid bilayer. All right, and this section right here, the nucleocapsid, is going to essentially form. Its membrane is going to come from essentially using a section of the membrane of the endoplasmic reticulum. All right, so you can see that when it's essentially like, you can picture this part right here pushing into the endoplasmic reticulum, and when that happens, it's going to pinch off a section of this bilayer right here, making this part right here, and that is essentially a new virus that's waiting to emerge from the infected cell. All right. There's then going to be a mechanism by which it's going to form what's called a Golgi apparatus, or Golgi vesicle, which is going to allow it to get to the surface of the infected cell, and that's eventually going to lead to what the process called exocytosis, when it's essentially you're releasing new virus particles. All right, so this is a simplified description of this. One thing to remember is that if you were to look at this, this would make it look like you only get one virus from one virus particle here. So you're actually going to be able to generate many virus particles, because the same positive strand that comes from one virus particle, this process can happen many times. So you can end up generating many new virus particles from a single virus. All right. In fact, if you only were able to create one virus, new virus from an old virus, it would be impossible for that virus to survive and proliferate. All right. You need to have, in order for this to be a successful virus, you need to be generating many new viruses for every one infected cell.